Knowledge changes lives. Welcome to Academic Deep Dive.
Hi everyone, welcome to our first deep dive into the engine room of the cell. Today we're starting our journey with glycolysis. If you've ever wondered how that pasta dinner actually turns into the energy you use to run or study, well, this is exactly where it begins. By the end of this explainer, you'll understand why your body literally traps sugar inside its cells and the absolutely fascinating difference between how your brain and your liver handle glucose. Let's get started. Here is our agenda for today. We'll start with part 1, the engine room, glycolysis, then move into part 2, phases of the pathway. After that, it's part 3, reaction 1, the trap, part 4, hexokinase versus glucokinase, and finally part 5, test your knowledge. Okay, moving right into part 1, the engine room, glycolysis. So glycolysis, which is also known as the Embden-Meyerhof-Parnas, or EMP pathway for short, is the universal sequence of reactions used by all tissues to oxidize glucose. Think of it as the ultimate foundation of metabolism. Its primary goals are basically twofold: providing energy in the form of ATP, and creating intermediates that other pathways can use as building blocks. Now, this entire process happens right in the cell's cytosol. It's a sequence of 10 specific reactions that take one 6-carbon glucose molecule and break it down into two 3-carbon molecules called pyruvate. And along the way, we net some ATP and a really important high-energy electron carrier called NADH. Now, pyruvate's journey from here completely depends on your environment. In aerobic conditions, meaning with oxygen, that pyruvate heads straight to the mitochondria to become acetyl-CoA for the TCA cycle. But in anaerobic conditions, say you don't have enough oxygen because you're a sprinting muscle, it's reduced to lactate instead. And what's really cool is that some microorganisms like yeast take a third path called alcoholic fermentation, turning that pyruvate into ethanol and CO2. It's just so adaptable. Let's look at how this is structured in part 2, phases of the pathway. We can actually split these 10 steps into 2 distinct stages. The first 5 steps are what we call the energy investment phase. Think of this exactly like a business startup. You have to spend money, in this case ATP, to make money later. We use ATP to phosphorylate our intermediates. This is then followed by a cleavage phase right in the middle, where we literally snap the molecule in half. Then the final 5 steps are the energy generation phase. This is where we finally see a return on our investment, gaining a net of 2 ATP per glucose molecule through something called substrate-level phosphorylation. We spent 2, we made 4, so we walk away with a neat profit of 2 ATP. All right, let's zoom in on that first crucial step. Part 3, reaction 1, the trap. Let's look closely at reaction 1. As soon as glucose enters the cell, we immediately phosphorylate it to glucose 6-phosphate, or G6P for short. Imagine a 6-sided ring, which is our glucose. An enzyme reaches in, takes a phosphate group from an ATP molecule, turning it into ADP, and just sticks that phosphate right onto the 6th carbon of the glucose ring. So why do we do this? Well, 2 big reasons. First, phosphorylated sugars are highly polar and bulky. They just don't have a transport protein to let them back out, and they definitely can't diffuse through the lipid membrane. By adding that phosphate, we effectively trap the sugar inside the cell and commit it to being metabolized. It's the Hotel California of biology, right? Glucose can check in, but it can never leave. And the enzymes that do this are incredible. Let's look at them in part 4, hexokinase versus glucokinase. This first reaction is so important that we actually have completely different enzymes for it, depending on where you are in the body. Most of your tissues use hexokinase. Now, hexokinase is ubiquitous, meaning it's found everywhere. It has a very low Km, which translates to a high affinity for glucose. So even if your blood sugar is super low, hexokinase acts like a vacuum. It will grab whatever glucose is available so your brain and muscles can keep functioning. However, it has a low Vmax, or maximal velocity, meaning it won't take more than the cell actually needs. It's also inhibited by its own product, G6P. So if the cell's full, the enzyme just shuts off. Now, the liver and the pancreatic beta cells are a completely different story. They use glucokinase. Glucokinase has a high Km, meaning it has a low affinity for glucose. It only really gets to work after you've eaten a huge carbohydrate-rich meal, when glucose levels in the portal vein are sky high. But it has a massive Vmax, allowing the liver to rapidly clear that flood of glucose from the blood to prevent hyperglycemia. And unlike hexokinase, it is not inhibited by G6P. Instead, it's inducible. Insulin actually tells your liver to make more of this enzyme. And in the pancreas, it acts as a clever glucose sensor to tell the entire body when to secrete insulin. To remember this difference for exams, just think of hexokinase as a greedy toddler. It grabs any glucose it sees immediately because of that high affinity, but it gets bored quickly, representing the low Vmax. On the other hand, think of glucokinase as a professional warehouse worker. It waits patiently for a big shipment to arrive, that's the low affinity or high Km, but then it clears the entire truck at lightning speed to keep the blood clean, which is that high Vmax. Let's wrap things up with part 5, test your knowledge. Go ahead and take a screenshot right here to test yourself on these core conceptual questions. Why is glucose phosphorylation a commitment step? How does the glucose sensor function of glucokinase in the pancreas relate to insulin secretion? Explain why phosphorylated sugar molecules cannot diffuse across the plasma membrane. Contrast the feedback inhibition mechanisms of our toddler hexokinase and our warehouse worker glucokinase. And lastly, why is it physiologically advantageous for the liver to have an enzyme with a high Km for glucose? If you can answer these, you're in fantastic shape. Finally, I'll leave you with this basic recall question to really cement what we've learned. In which specific compartment of the cell does this entire glycolytic process happen? Think back to our very first concepts. If you don't know the answer right away, no worries. Just scrub back to the beginning of this explainer. Thanks so much for joining me on this journey into cellular metabolism. Keep studying hard, and I'll catch you in the next one.
Welcome back. In our last explainer, we saw how the cell trapped glucose by turning it into glucose 6-phosphate. But, trapping it is really just the beginning. Today, we're looking at how the cell decides to actually burn that sugar for energy. We're covering the point of no return in glycolysis and the incredible way your body uses hormones like insulin and glucagon to control this entire process. Let's dive into reactions 2 and 3.
Okay, let's dive into this. Here is our roadmap for today. We'll start with the setup step. Reaction 2. Then, we cross the point of no return. After that, we'll talk about regulating the master enzyme, introduce hormones, and a massive super-activator, give you a really sticky factory memory aid, and wrap up by acing some exam questions.
Section 1. The setup. Reaction 2.
So, we have glucose 6-phosphate sitting right there in the cytosol. Reaction 2 is basically a quick shuffling step. An enzyme called phosphohexose isomerase, sometimes called phosphoglucose isomerase, takes that glucose 6-phosphate and turns it into fructose 6-phosphate. Just imagine our standard 6-carbon glucose ring. The enzyme essentially rearranges the atoms to turn it into a 5-sided fructose ring. But it still has 6 carbons total, and that phosphate group, it stays exactly where it is.
Now, why bother with this? Well, this reaction is readily reversible and honestly isn't a control point at all. Its main job is just to prepare the molecule for the next step by moving the carbonyl group around. That makes it so much easier for the cell to snap this molecule in half later on. And because it's totally reversible, the cell can easily move back and forth between these two sugars depending on what else is happening in metabolism.
Section 2. The point of no return.
Now we hit the big one. Reaction 3 is what we call the committed step. In biochemistry, a committed step is an irreversible reaction. Once a molecule passes through this gateway, it is 100% committed to finishing the entire pathway. There is no turning back.
This reaction is the phosphorylation of fructose 6-phosphate into fructose 1,6-bisphosphate, and it's catalyzed by the absolute master regulator of glycolysis, phosphofructokinase 1, or PFK-1. This is the most important control point in the entire pathway. The cell actually spends its second molecule of ATP right here, so it's making a real tangible investment. It's exactly like clicking Confirm Purchase on a non-refundable ticket. You're going on this trip.
Section 3. Regulating the master enzyme. Because PFK-1 is the gatekeeper, the cell regulates it with extreme care, using allosteric regulation. Basically, molecules bind to the enzyme and flip its on or off switches. Let's look at the off switches. If the cell is already rich in energy, it just doesn't need to burn more sugar. So high levels of ATP act as an energy-rich signal and inhibit PFK-1. Another big inhibitor is citrate. Citrate is the first product of the TCA cycle, which happens over in the mitochondria. If citrate levels are high, it tells the cytosol, hey, the mitochondria are full, stop sending us more fuel. Conversely, if the cell is starving for energy, it produces AMP. High AMP tells the cell its energy stores are depleted, and it strongly activates PFK-1 to get the energy production line moving.
So you have this constant push and pull. You've got ATP and citrate actively trying to slow the enzyme down, while AMP is giving it a massive green light. But there's actually another activator in the mix, and it leads us to one of the most fascinating mechanisms in human biology.
Section 4. hormones, and the super-activator.
Okay, there is a super-activator you absolutely need to know for your exams. Fructose 2,6-bisphosphate. Notice the 2,6 there. This is completely different from the fructose 1,6-bisphosphate we just made in glycolysis. Fructose 2,6-bisphosphate is not a glycolytic intermediate. It's a dedicated, specialized signaling molecule, and it is the single most potent activator of PFK-1.
Here is how your hormones actually use it. Think about the well-fed state. You've just eaten, so insulin is high and glucagon is low. This hormonal environment activates a totally separate enzyme called PFK-2 by dephosphorylating it. Active PFK-2 creates a massive flood of fructose 2,6-bisphosphate, which then violently kicks our master enzyme PFK-1 into high gear. The result? Your liver rapidly burns glucose. But what about the fasting state? You're hungry. Glucagon is high. Glucagon triggers the phosphorylation of PFK-2, which actually inactivates it. That causes your fructose 2,6-bisphosphate levels to drop. Without its favorite activator, PFK-1 shuts down, stopping glycolysis in the liver so that the liver can instead make glucose for the rest of your body.
This brilliant setup prevents a biological paradox known as a futile cycle. A futile cycle is when a cell is trying to burn glucose through glycolysis and simultaneously trying to build glucose through gluconeogenesis. You'd just be wasting energy. Fructose 2,6-bisphosphate prevents this perfectly because while it highly activates glycolysis, it simultaneously inhibits gluconeogenesis. It is the ultimate master toggle switch.
Section 5. The factory memory aid.
I know, this is a lot of biochemistry to keep straight. So think of PFK-1 as the lead engineer of a factory. ATP and citrate, those are just the stop memos coming down from the warehouse saying the inventory is full. AMP is the flashing emergency alarm saying the power is out and they need to work frantically. But fructose 2,6-bisphosphate, that represents the big boss, insulin, actually walking onto the factory floor. When the boss is in the room, the lead engineer ignores everything else and works at 200% capacity. Keep that factory in your head and you won't forget these regulators.
Section 6. Ace your exam questions.
Let's do a rapid-fire review to validate what you've learned. First, a conceptual question. Why can the isomerization of glucose 6-phosphate to fructose 6-phosphate occur in either direction, while the PFK-1 reaction absolutely cannot? Well, reaction 2 is just a preparation step. It shuffles atoms around without a big energy investment. But reaction 3 consumes an ATP to lock the molecule into the pathway as an irreversible, non-refundable, committed step. Next question. Pure recall, but absolutely vital. Which molecule is the single most potent activator of PFK-1?
It is fructose 2,6-bisphosphate. Do not forget that 2,6. And remember, it's created by PFK-2.
Let's finish with a tough conceptual question. How exactly does fructose 2,6-bisphosphate prevent a futile cycle? It acts as a master toggle switch. It activates glycolysis through PFK-1 while simultaneously inhibiting gluconeogenesis. It ensures the cell is literally never working against itself.
As we wrap up this explainer, I want to leave you with this thought. Isn't it absolutely incredible how a single microscopic signaling molecule can totally dictate whether your body aggressively burns or carefully saves its energy? It's evolutionary biochemistry at its absolute finest. Thank you so much for learning with me today, and I will see you next time as we tackle the rest of the glycolytic pathway.
Welcome back to the explainer. So, in our previous lessons, we spent 2 ATP molecules just to prime our glucose, turning it into fructose 1,6-bisphosphate. You can think of the molecule at this stage kind of like a fully loaded spring, just ready to snap. And today, we finally get to the cleavage phase. We're going to take that 6-carbon sugar and split it right down the middle. Literally, this is the exact moment where the glyco, meaning sugar, meets lysis, meaning splitting. Let's dive right into reactions 4 and 5. We already invested the energy to prime our glucose, but what actually happens when you snap that fully loaded molecular spring in half? We're shifting from just prepping our molecule to actually breaking it apart. Let's see exactly how this cell meticulously handles this breakdown. Okay, picture our 5-sided fructose ring from the last lesson. Notice how it now has a bulky phosphate group sitting on each end, specifically on carbons 1 and 6. This is fructose 1,6-bisphosphate, and it is perfectly primed for the great split we're about to witness in reaction 4. Enter aldolase. This enzyme's job is straightforward, but absolutely critical. It acts just like a pair of precise molecular scissors, making a cut right between carbon-3 and carbon-4. Now, definitely note this for your exams. This specific cleavage reaction is entirely reversible, and unlike those earlier investment phase enzymes we talked about, it is not a regulated step. And here's the brilliant result of that cut. The scissors do their job, but the two resulting pieces, they aren't identical twins. They are structural isomers. One is dihydroxyacetone phosphate, or DHAP, and the other is glyceraldehyde 3-phosphate, or G3P. See how each of these 3 carbon molecules walks away with its very own phosphate group? To easily remember these 2 products for test day, just think of them as the DAAP and the GAP. The GAP, glyceraldehyde 3-phosphate, is literally the gap that the rest of the pathway can smoothly walk right through. The DAAP, on the other hand, operates a bit more like a side door, but usually it just gets tapped back into the main hallway by an isomerase, so it can be burned for energy. Now, here is a fascinating clinical nuance you will definitely see on board exams. While general tissues use standard aldolase for cellular glycolysis, the liver relies on a highly specific isoform called Aldolase B. Why does that matter? Because Aldolase B is also tasked with handling your dietary fructose metabolism. So if you ever come across a clinical vignette about a patient with fructose intolerance, Aldolase B is the exact enzyme you need to recall. But returning to our main pathway, we hit a massive problem. The rest of our glycolytic machinery is strictly built to only process the GAP. That means the DAAP molecule we just made is essentially a metabolic dead end for energy production. Well, unless we can somehow change its shape. And that brings us perfectly to reaction 5, the final step of the cleavage phase. We absolutely have to make the pieces match to keep the machinery moving. I mean, a biological system simply cannot afford to just leave half of its hard-earned glucose investment sitting there doing nothing, right? So in steps triose-phosphate isomerase, or TPI. This enzyme readily interconverts the DHAP and the G3P, and because the cell is constantly pulling G3P away to feed the rest of the glycolytic pathway, the equilibrium naturally shifts. That constant removal effectively forces all of that remaining DHAP to turn right into G3P so it can actually be utilized. Here is the crucial point, and it's a massive paradigm shift. By the end of reaction 5, one single molecule of glucose has effectively become 2 molecules of glyceraldehyde 3-phosphate. This is literally the most important mathematical takeaway of today's lesson. From this point forward, everything in the biochemical pathway happens twice for every 1 glucose molecule we originally started with. Let's step back for a second and look at the larger metabolic picture. Why doesn't the cell just throw the DHAP away if it just has to be converted anyway? Well, because biology is brilliantly efficient. The DHAP isn't just some backup, it has its own side hustle. DHAP acts as a side door to synthesize glycerol 3-phosphate, which serves as the critical backbone for triacylglycerol, or TAG synthesis. It's a beautiful example of how glycolysis provides the exact building blocks needed for other cellular functions, directly linking energy breakdown to fat storage. All right, bringing it all together, here is our overarching narrative for this phase. We primed our asset using ATP. Then, we used aldolase to snap our 6-carbon sugar into 2 3-carbon pieces. Finally, we forced those pieces to match using triose-phosphate isomerase. We now have 2 highly functional G3P molecules fully ready for processing. Now, let's make sure this sticks. We're going to run through some rapid-fire recall and conceptual questions you'll definitely face on exams. First up, do you remember the name of our molecular scissors? The enzyme responsible for splitting fructose 1,6-bisphosphate is, of course, aldolase. Next, think back to that factory limitation we discussed. Why is it absolutely necessary for the cell to utilize TPI to convert DHAP into G3P? Well, it's because the subsequent enzymes in glycolysis are incredibly specific. They simply cannot bind or process DHAP. Without that isomerization step, half of the potential energy from our original glucose molecule would just be completely wasted. Okay, how about that DHAP side hustle? What exactly is the link between the cleavage phase of glycolysis and the synthesis of fats? Remember, DHAP can be diverted away from glycolysis to synthesize glycerol 3-phosphate. That provides the indispensable backbone for synthesizing triacylglycerols, which, you guessed it, are your stored fats. Let's try a clinical application question now. If a patient has a defect in aldolase B, which specific dietary sugar will cause them the most trouble? As we covered earlier, while general aldolase handles glycolysis everywhere, aldolase B in the liver specifically manages dietary fructose metabolism. So the correct answer here is fructose. And finally, let's lock in the math of glycolysis. How many molecules of glyceraldehyde 3-phosphate do we ultimately yield from a single glucose molecule? The answer is 2. Because of that doubling effect after reaction 5, every single subsequent energy-yielding step has to be multiplied by 2. So we finished the energy investment, and we've completely split our molecular assets. But wait a second. If glycolysis is supposed to produce energy, and so far we're sitting at a net negative of 2 ATP, when do we actually get paid? In the next phase, how exactly will the cell finally start cashing in those 2 molecules of G3P to reap a massive energetic reward? Think about how cellular oxidation might play a role here, and join me in the next explainer to find out.
This has been Deep Dive Academy. Learn deep, remember forever.